Hi, so I'm John Inadomi. I'm the division head of GI, and I'm very pleased to introduce our speaker for the Wade Vollweiler Visiting Professorship. It's Dr. Rick Peek. I'll talk to him about him in a second, but Rick is a professor of medicine and the Mina Cobb Wallace Professor of Medicine, Cancer Biology, Pathology, Microbiology, and Immunology. Like, wow, all the courses I did do well in med school. And currently the director of the division of GI at Vanderbilt. But a little bit about this particular series. The Wade Vollweiler was the first division head of GI. Oh, do I need to speak louder? Okay. He was the first division head of GI. He was born in Grand Forks, North Dakota, attended Oberlin College, received his MD from Harvard, and he did his residency training at Mass General, and research training at the Mayo. Recruited to UW in 1949, and then was a division head for the next 31 years. His research interests were quite varied. They're both in hepatitis, as well as luminal disease, celiac disease, and malabsorption. And became the president of three major societies. One is the American Gastroenterological Association, which is our big GI society, as well as the American Association for the Study of Liver Disease, which is the biggest liver society. And also the Western Association of Physicians. And in fact, during that time, he received the Friedewald Medal, which is the highest award that the American Gastroenterological Association can give on to a member. So that's who we're honoring. But I'm very pleased to announce that Rick Peek is our speaker this year. Rick got his medical degree from University of North Carolina in Chapel Hill, internal medicine residency at University of Alabama in Birmingham, and his GI fellowship at Vanderbilt. He was then recruited to stay at Vanderbilt as an instructor, and then progressed as an assistant, an associate, and a full professor, and has been the director of the GI division since 2004. Rick has a very well-established research program, very well known for his work in Helicobacter pylori and its effect on GI biology, inflammation, and gastric cancer. His work has really been fundamental in understanding the strain-specific genetic abnormalities that are associated with inflammation causing gastric cancer as well as peptic ulcer disease. And basically who gets cancer and who gets ulcers. But conversely, some of his more recent stuff isn't dissing H. pylori the whole time. In fact, H. pylori at certain strains may be protective against Barrett's esophagus and esophageal adenocarcinoma. So the whole picture is quite interesting. Most recently, actually, I was noticing they also looking at the role of gastrin and colon cancer, which of course is something I'm quite interested in. Rick has over 200 publications in peer-reviewed journals. His research funding is quite extensive. He's the PI of at least 5 NIH grants, including a couple R1s, a PO1, P30, and a T32 training grant. He's had a whole bunch of international speaking engagements. We're really happy to have him here. I've had at least 30 countries on my list of places you've visited. And as it turns out, he was in Seattle last about 2004. You can talk about that. It probably was a H.pylori conference that was, or at least I think it was a vaccine and prevention symposium that involved H. pylori at that time. He's mentored 45 fellows and faculty members, and he's had a whole bunch of national honors, including election to the American Society of Clinical Investigation, awards from our major GI societies, including the American Gastroenterological Association. I met Rick when he was the chair of AGA Council. AGA Council is the body that's responsible for the educational component of the American Gastroenterological Association and responsible for putting together the educational programs for the AGA. And now he's the editor-in-chief of the journal Gastroenterology. And under his leadership actually has its highest impact factor. It's up to 20 now, which is pretty impressive for a subspecialty journal. So this week, as the part of the Vollweiler lecture series, tonight I'm telling you a little plug at our Pacific Northwest GI Society. We'll be talking about optimizing management of H. pylori in 2018. Tomorrow at our GR Frontiers at 7:30, if you want to come, he's going to be talking about these relationships to promote development of gastric cancer and H. pylori. And today, for our department grand rounds, he's going to give a talk on H. pylori-induced gastric cancer, tragic triangulations. So Rick, thanks so much for visiting Seattle, and I really look forward to your talks.
Well, Well, John, thank you so much for that incredible introduction. I wish my wife were here to hear that. Yeah, right. I hope you recorded that. I also wanted to express what a tremendous honor it is for me to be here in recognition of such prolific individuals, Dr. Vollweiler. I had the opportunity to read in Gastroenterology the address where he received the Friedenwald Medal from the AGA, and he was indeed an incredible scientist, administrator, researcher, and clinician. The broad swath of topics he was an expert in really, makes him one of the giants in gastroenterology. I also wanted to say that there are incredible and even striking parallels between Vanderbilt and the University of Washington. One of our first division director at Vanderbilt was Dr. Harrison Schull, and he trained at MGH. He trained with Chester Jones, who Dr. Vollweiler also trained with, and I'm sure that they were colleagues. And in fact, our signature lectureship, the Schull lecture, John has given that, Dr. Lee has given that. I have great friends who went through Vanderbilt or we recruited from Seattle on the faculty, and in fact, right now, Dr. Bradley Annawalt from your institution is giving medical grand rounds at Vanderbilt. So we're in these parallel universes. So I wanted to thank you again for the opportunity. And what I'm going to do this morning is talk about some of what I consider to be dysfunctional relationships between H. pylori and its host that really seem to predispose to gastric cancer. So essentially all persons who are colonized with H. pylori develop coexisting gastric inflammation. And over a period of decades, this is the strongest known risk factor for distal gastric cancer. And in fact, the attributable risk conferred by H. pylori on gastric cancer is about 90%. This is a lesion that I identified a couple of years ago at a patient at the VA. This is in the distal portion of the stomach. It's very irregular mass lesion. It was very friable when we biopsied it, and it turned out to be an intestinal type gastric cancer. Worldwide, gastric adenocarcinoma is a big problem. It's the third leading cause of cancer-related death. There are over 780,000 deaths per year, and 1 million new cases of gastric cancer will be diagnosed this year. We know a lot more now about the molecular features of this malignancy, thanks to work by the Cancer Genome Atlas Network. They used a multitude of techniques to molecularly classify gastric adenocarcinoma into 4 different categories: chromosomal instability, those caused by EBV, microsatellite instability, and genomically stable malignancies. But in every part of the stomach, chromosomal instability type tumors are the most predominant. And this is strongly linked with a type of gastric cancer called intestinal type gastric cancer. We know very well from studies pioneered by Pelayo Correa that intestinal type gastric cancer progresses through a very well-orchestrated cascade of histologic lesions, from normal mucosa to non-atrophic gastritis and all the way to intestinal type gastric cancer. And H. pylori initiates this carcinogenic cascade. So many studies have now tried to demonstrate that if you eradicate the bacteria, you can lower the risk of subsequent development of gastric cancer. So some of the earlier studies actually were a little disappointing. This is a randomized controlled trial from China by Benjamin Wong, in which they randomized patients into those that received placebo and those received antibiotics targeting H. pylori. And they followed them for about 5 to 7 years. And although there did seem to be a difference, this was not statistically significant. And really the only benefit that they identified was when they did a post hoc analysis and they looked at individuals who did not have pre-malignant lesions. And in those individuals, it did look like that treatment of H. pylori could lower the risk of developing gastric cancer. So why did this large trial fail to identify an overall benefit? Well, I think it had to do with the length of follow-up. So this is a chronic disease that occurs over decades. And these are data from a study we published with Paulaya Correa in Colombia, in which we looked at individuals who had premalignant lesions. And all we wanted to see was whether the severity of these premalignant lesions would change if patients were treated for H. pylori and successfully eradicated, if they were treated but had persistent infection, or if they were untreated. And it took almost 12 years to identify any significant reduction in the intensity of premalignant lesions. So I think that longer follow-up is a key to looking at studies of gastric cancer within the context of H. pylori. And indeed, now we have very good randomized controlled trials that have shown that if you treat H. pylori and follow patients over 15 years, you will see a significant reduction in the development of gastric cancer. Now, there have been many trials. These are 4 of the first prospective long-term trials that have shown if you pool these ratios, there is a significant reduction in the risk. There was also a very interesting article published this year in the New England Journal looking at premalignant lesions and early-stage gastric cancers in the stomach. And this was performed by a group in Korea. They identified patients endoscopically that had gastric adenomas or early-stage gastric cancer. Those patients had these lesions endoscopically resected, and then half were treated for H. pylori, half were treated with placebo. And they found that following these patients over many, many years, that there was a significant difference in the development of metachronous gastric cancers in the H. pylori treated group. And further, when they looked at people that were actually successfully treated, who had documented eradication of H. pylori, this difference was even more marked. And so now many studies have been pooled together in meta-analyses. This is one that we published in Gastroenterology to show that indeed there is a significant benefit of treating H. pylori for the development of gastric cancer. But it's interesting because the effects are more pronounced in populations that have higher background incidence rates for this malignancy. So why don't we test and treat everybody for this carcinogen? Well, one reason is that only a minority of the billions of people that are infected worldwide will ever develop gastric cancer. So I think that this has forced investigators to try and understand the determinants which may influence who is colonized that will progress the disease and who remains asymptomatic. So strain variation has been an intense area of focus for our particular group. Many studies have now shown that H. pylori is a very genetically diverse pathogen. These are data from a study that was published in Nature, in which the investigators looked at over 700 H. pylori strains. And they sequenced certain genes in those strains, and then they compared them. And they found that the strains actually began to fall into clades, and there were about 9 different groups or clades of strain classifications that fell out. And they very much paralleled the phylogeographic origin of the hosts from which those strains were obtained. And so H. pylori now has been used as almost a poster child to trace populations of humans because it's been associated with humans for over 200,000 years. And in fact, there's a prevailing theory that populations in Europe, human populations, actually formed by a fusion of migration from individuals from Asia and Northeast Africa. And this is postulated to have occurred about 50,000 years ago. But now studying the H. pylori strains from those patients, new parameters and new hypotheses have been raised. Specifically, one of the more interesting articles that focused on this was looking at an H. pylori strain isolated from a very famous mummy that was frozen in the Italian Alps called the Iceman. And carbon dating of the Iceman showed that he was about 5,300 years old. This is a picture of the Iceman. They've named him Otzi. They've reconstructed him, and they think he looks like Harvey Keitel. And the investigators in this study, they used techniques to amplify ancient DNA, and they were actually able to amplify H. pylori specific sequences from the putative stomach of the Iceman. And they compared that to all these other strains. Now, if the prevailing theory is correct, that this population resulted from a fusion of these two populations 50,000 years ago, then you would expect the Iceman strain to be right here. But in contrast, it was not. It was a pure Asia 2 strain. And so that suggests that this fusion population actually occurred much later than population biologists originally speculated. So how can we use strain-specific virulence to understand carcinogenesis? Well, one of the molecular proteins that H. pylori synthesizes is a protein called CAG-A. And about 60% of H. pylori strains harbor this protein. And when the bacteria bind to epithelial cells, this protein is injected into host epithelial cells. It interacts and aberrantly activates many different pathways, all of which have been shown to be mutated in gastric cancer. If you put CAG-A into a host cell, shown in green, and watch the behavior of this cell in a monolayer, it migrates much farther and much faster than other cells that surround it. So the intracellular presence of CAG-A confers carcinogenic phenotypes on epithelial cells. One of the more interesting areas of research now has been how do carcinogenic strains of H. pylori interact with susceptible populations in the stomach, namely progenitor or stem cell populations. And there was an incredible study by my very good friend Manuel Amieva at Stanford, in which they infected animals with H. pylori. They used very innovative imaging techniques. This is confocal in vivo microscopy. They used full thickness biopsies of the stomachs from these animals. And using this, they began to identify pockets of replicating H. pylori that were not really appreciated before. And these were deep down within the stem cell regions of the gastric glands. And they found that cells that were adjacent to these populations of H. pylori were undergoing mitosis and increasing in proliferation. So it seems that these intracellular replicating colonies of H. pylori were driving proliferation in the stem cell zone. They also showed that H. pylori could activate these stem cells. These are lineage tracing studies in which you can mark a stem cell population, and then over a period of time you can follow the marked cells, showing the cells that emanate from the stem cells. And this is what happens in a normal uninfected stomach. You see some glands that are populated by stem cells and daughter cells from those stem cells. But when you infect these animals with H. pylori, you see a marked increase in the activity of these stem cell populations. What about host genotypes? Do they predict or can be potential biomarkers for people that are at higher risk for gastric cancer? There was a very good GWAS study that was published in JAMA a few years ago, in which they looked at two populations in Europe, and they looked for particular genetic loci which were associated with H. pylori positivity. And using this Manhattan plot, they identified two different loci with SNPs that significantly associated with H. pylori infection. One of these SNPs localized to an innate immune receptor called toll-like receptor 1. And this recognizes triacylated lipoproteins from gram-negative bacteria. And they found in both of these populations that the expression level of TLR1 paralleled the haplotype of this SNP. I think, future opportunity from this particular study that was raised is to now look at people that are infected with H. pylori who actually develop gastric cancer. This study did not actually do that. So now if you look at host genetic factors and bacterial factors in context, can that more accurately predict biological outcomes of infection? And we were very fortunate at Vanderbilt to be able to work with Dr. Correa's group. We recruited them up in 2005. They've been an incredible group of individuals to work with. Dr. Correa is a very famous pathologist. He, before the discovery of H. pylori, he was following populations in Colombia, and he's the one that delineated all the histologic steps to intestinal type gastric cancer. So he has many populations in Colombia they've followed now for over 45 years, and they found an incredible dichotomy that almost everybody in Colombia is infected with H. pylori. But the individuals that live on the coast have a much lower risk of gastric cancer than the individuals that live in the mountains, and it's a 25-fold difference. So what accounts for these differences if everybody in the country is infected? Well, you can look at the genetics, because there's samples from these individuals that are obtained every 3 years. We have the human DNA, we have the H. pylori strains that infect these individuals. And so Dr. Correa's group looked at the genetic background of the hosts that live on the coast versus the mountains and found that there were significant differences. They also looked at the genetic diversity of the H. pylori strains that were isolated from those individuals. Again, they're different. But what if you look at them together? So in this slide, the top two panels are the genetic diversity of the host from coast and mountains. The bottom two slides are the genetic diversity of the strains, coast and mountains. And each white vertical line that's extending down across the panels is the genetic diversity of one person, followed by the genetic diversity of their H. pylori strain. And immediately you can see that these patterns are not the same. So does genetic discordance predict disease? And in fact, it does, at least in this population. So this is a heat map looking at the severity of lesions within the stomach, ranging from gastritis alone to gastric dysplasia. And in individuals that have a high proportion of Amerindian ancestry, but who are infected with H. pylori strains with a high proportion of African ancestry, all of these individuals had gastric dysplasia. You can look at it another way. In individuals who have an African ancestry infected by an African strain, their mean lesion is non-atrophic gastritis. Amerindians who harbor Amerindian H. pylori strains, their mean lesion is mild gastric atrophy. When there's a genetic mismatch, the mean lesion is severe intestinal metaplasia. So what about environmental cofactors? This has also been an area that we're really interested in at Vanderbilt. Epidemiology studies have consistently shown that high salt diets predispose to gastric cancer. How does this happen? Is it simply mucosal damage that's incurred over a period of decades due to exposure to these diets? Well, at Vanderbilt, one of my colleagues, Dr. Tim Cover, has looked at the effect of high salt on the strains of H. pylori. And he's found that the expression of this CAG-A oncoprotein is markedly increased in high concentrations of salt, raising the hypothesis that dietary elements can manipulate the virulence potential of this carcinogen. So our lab has been really interested in iron deficiency. So iron regulates microbial virulence. H. pylori infection is a very strong inducer of iron deficiency. And in fact, iron deficiency is present in about 30% of the world's population. And finally, iron deficiency is associated with premalignant and malignant lesions in the stomach. So we did a very simple experiment using our animal model of H. pylori. We took gerbils that were fed a normal diet or that were fed an iron deficient diet. We then infected these two groups with H. pylori, and we looked for disease. And we found that the frequency of dysplasia in yellow and carcinoma in red was twice as frequent if the animals were iron deficient when we infected them with H. pylori. How did this happen? Well, again, we returned to this CAG-A oncoprotein. And we found that when we took H. pylori strains from gerbils that were iron normal, you add them to gastric epithelial cells, these secretion pili that are seen as these threads on this electron micrograph begin to form. But when you take an iron deficient strain and add it to epithelial cells, there are many more of these secretion pili that are formed, translocating this oncoprotein. Well, an important aspect of our lab has always been to try and validate what we see in the lab, ex vivo, in animal models, in human populations. So again, we work with Dr. Correa's group. We were able to show that in individuals from Colombia who had the lowest ferritin concentrations, they're the ones that had the higher severity of disease in the stomach. But we had their strains too. And when we looked at the strains from patients who had the lowest iron levels, they were able to induce much more of a pro-inflammatory response than strains from patients with high ferritin levels. So, because I trained with Dr. Blazer, I always have to think about things from the bacteria perspective. So why would a bacterial pathogen that colonizes for decades want to eliminate its host? Well, we did some studies, again, with Manuel's group at Stanford to identify some clues for this phenomenon in phenotype. So they had a polarized cell system in which you can add H. pylori in green to epithelial cells. And when you do that, H. pylori grows very well, it forms microcolonies. If you knock out that CAG-A oncoprotein, the bacteria cannot survive on this epithelial monolayer. But if you add iron back to this mutant, all of a sudden the bacteria can grow again, suggesting that one function from the bacterial perspective for CAG-A is to acquire iron or an iron source from its host. So Manuel did a lot of work to try and define this mechanism, and basically the upshot of their results was that if you add H. pylori to epithelial cells, it injects CAG-A into the host cell. And one of the consequences is that transferrin, an iron source, is moved from the basolateral surface of the cell to the apical surface of the cell, where it now supplies an iron source for the bacteria to grow and replicate. And consequently, if you put the bacteria under iron deficient conditions, its response is to inject more CAG-A into the host cell to get more iron. But a detrimental consequence long-term, especially if it's affecting a susceptible stem cell population, is an increased risk for carcinoma. So I think looking at all these elements in context is very important. I think previously as investigators we've categorized very conveniently microbial constituents, host effectors, environmental cofactors into very neat categories, but we've understood now that we really have to define how they interact with each other to really understand the combinatorial risk they exert on the development of gastric cancer. So I wanted to talk about some more exciting areas that we're looking at in the future, and also bring up some clinical questions that we're faced with, when dealing with individuals who are colonized with this bacteria. So I showed this slide earlier. This is the slide from a study in China where they treated for H. pylori. They were able to show that there was a significant reduction in the incidence for gastric cancer. But buried deep within this article was the fact that 15 years after they treated those individuals that still showed a benefit for reducing the incidence of gastric cancer, almost half of them were still infected with H. pylori. So this raises the question, are there roles for non-H. pylori constituents of the gastric microbiota in disease outcome that we have to think about? So we know a lot less about the gastric microbiome than the intestinal microbiome, but one thing is clear from clinical studies. If you look at humans, in the stomachs of individuals who are infected with H. pylori, this organism predominates. There's much less diversity of microbial populations in the stomach of H. pylori infected individuals compared to H. pylori uninfected individuals. In mice, you can see the same phenomenon. This is a classification of the microbiome in uninfected mice. If you infect these mice with H. pylori, you see dramatic shifts. You see an expansion of the firmicutes population. You see a contraction of the Bacteroidetes. Does this have anything to do with disease? Well, Jim Fox and Tim Wong had a study that suggests indeed it does. So this is a study in which they took germ-free mice compared to normally raised mice, conventionally housed mice. And they infected them with H. pylori. And they wanted to see if there were any differences in the development of neoplasia. And they found that in animals that were germ-free, that were monocolonized with H. pylori, the majority of these mice developed no evidence of neoplasia at this time point. And the ones that did only had low-grade lesions. In contrast, mice that had a normal microbiota that were infected with H. pylori developed a much higher risk of neoplasia. And the majority of these lesions were high grade, suggesting that there's active collaboration between H. pylori and its gastric microbial constituents. There are also human data to suggest that the microbiota may be important. So this is a study published by a computational biology group. And they developed a technique in which they could query known cancer genome databases. And what they were looking for is evidence of bacterial DNA that had been integrated into the cancer genomes. And they found that the malignancy that had the second highest number of bacterial lateral genetic integrations was gastric cancer. But it was not H. pylori DNA that they identified. The highest bacterial species was Pseudomonas. So I think all these data have forced us to rethink a model of H. pylori-induced gastric cancer. I think early on, there are intense interactions between H. pylori and its host. There's a profound inflammatory infiltrate. However, over periods of years to decades, this interaction diminishes, gastric atrophy develops, and later there's colonization by different microbial species that may be important in driving these later stages of carcinogenesis. So what are these bacteria? Again, we can go back to these different populations and compare the microbial constituents in people at low risk versus high risk. And we've been able to do that with Jim Fox and Sebastian Serabom. And we found that there are bacteria that are only present in the low-risk individuals that are not present in the mountains. And there's even a smaller number of bacterial species that are present in the high risk versus the low risk. So now this has given us a template for us to do studies in humanized mouse models in which we can monocolonize with these different species and see if they affect the risk of progression to gastric cancer. So let's move into some clinical discussions. You know, I mentioned earlier that only a minority of individuals will develop gastric cancer who are infected with H. pylori. Are there other reasons that make us think twice about indiscriminately testing and treating for this bacterial species? Well, John mentioned some of the work that we were fortunate to be involved with, looking at the relationship between H. pylori and esophageal disease. I mean, I know Dr. Vaughn here is an incredible expert in esophageal adenocarcinoma. It has premalignant lesions that drive the progression to cancer, Barrett's esophagus. But many studies have now shown that there's a significant inverse relationship between the presence of H. pylori and premalignant and malignant lesions in the esophagus. This is just one of those studies published by Amnon Sonnenberg, showing that persons who are infected with H. pylori, persons who have gastritis, have a significantly lower risk of harboring Barrett's esophagus. This has also been extended to esophageal adenocarcinoma. This is from Wong Ho Chow, the National Cancer Institute, showing that indeed individuals that are infected with H. pylori, specifically strains that are CAG positive have a lower risk of esophageal adenocarcinoma. There have now been meta-analyses looking at this. And indeed, when you pool all these risks, there's a significant inverse relationship. It's not present when you look at squamous cell carcinoma, and it's specific for CAG positive strains, because when you look at the effect with CAG negative strains, it's no different. What about other diseases? Well, again, Dr. Blazer really has pioneered this area, looking at the relationship between H. pylori and autoimmune or atopic diseases. This is just one of their studies showing that if you look at the presence of H. pylori CAG positive strains on the risk for asthma, it's significantly reduced. I don't have time to talk about these other diseases, but all of these, in one way or another, have been inversely associated with the presence of H. pylori. So this begs the question, who should we test? Who should we treat? I think in 2018, this is the list that I would offer. Definitely individuals who have ulcer disease or who have gastric maltomas. If patients have a family history of gastric cancer, I would test and treat them. Patients who have a relatively rare disease, Menetrier's disease, because it's been linked to H. pylori. There's a synergistic risk between H. pylori and long-term non-steroidal use on the subsequent development of ulcers. There's also a small benefit of treating H. pylori in persons who have non-ulcer dyspepsia. Prior to long-term proton pump inhibitor use is a little bit controversial, but I think that the data do weigh in favor of testing and treating individuals who are going to be put on long-term PPIs for H. pylori and treating them. And then finally, as evidenced by the New England Journal article, any persons who have premalignant or malignant lesions in the stomach obviously should be tested and treated for this infection. What do we have in our armamentarium to treat H. pylori? Triple therapy has been used for many years. In some recommendations, it's still the very first line therapy. Quadruple therapy is now gaining a lot of momentum as first-line therapies. This can be in the form of bismuth-containing quadruple therapy, or it can be in the form of what we call concomitant therapy, which is a PPI and amoxicillin, clarithromycin, and metronidazole. So how do we decide which first-line therapy we should use? This is the cover of our very first journal issue as our team took over. And we were fortunate because we were able to publish guidelines reflecting the management of H. pylori in this first article. And these were the Toronto guidelines. And I think that these are very, very common sense guidelines because they approach things based on what you know about the local resistance pattern of your H. pylori infection in your community. And I'll be the first to tell you, I do not know what it is in Nashville. And many investigators and clinicians don't know what the resistance rates are for their H. pylori strains in their community to clarithromycin, which is really the important antibiotic to know the resistance patterns for. So the algorithms from these guidelines started with these questions. Do you know the local pattern of resistance in your community? If you do, that's great. And if it's low to clarithromycin, then yes, you can start with triple therapy. PPI, amoxicillin, and clarithromycin, and it goes through nice secondary regimens that you can use. If the person's allergic to penicillin, you can substitute metronidazole. But what if you don't know, like I don't know? Then this is what you should use. 4-drug therapy as the first line for 14 days. And it can be concomitant therapy, or you can start with bismuth quadruple therapy. And it gives nice choices if these therapies fail. So another question is, what if we do endoscopy and we identify a premalignant lesion in the stomach, atrophy or intestinal metaplasia? How do we follow those patients? What's the optimal surveillance protocols that they should be entered into? I'll tell you from the start, there's no good data on which to base these recommendations. And the AGA right now has a working group to actually propose management guidelines. These are guidelines that were published 4 years ago from Europe. I was involved in this process. And basically, if you have individuals that have gastric atrophy or intestinal metaplasia, you can stratify those individuals into those that have lesions only in the antrum or that have more extensive lesions in both the antrum and the corpus. In both of those groups, you should treat those individuals for H. pylori. If they only have these lesions in the antrum, you don't need to follow up those patients anymore. However, if they have more extensive lesions, then they should be followed up every 3 years with endoscopy with biopsies. So finally, what about the perspective for developing a vaccine? I will tell you that since the 1990s, there's been intense interest from pharmaceutical companies to develop an effective vaccine against H. pylori that will not only prevent disease, but there's also been interest in developing therapeutic vaccinations. Many of these trials did not really come through with very promising results until 3 years ago, when there was a large study published in China with over 2,000 children randomized to either group. There were very many strengths of this trial. Number one, it targeted populations, children, that were the most susceptible to acquiring H. pylori infection. Secondly, their primary endpoint was the development of endogenous H. pylori infection. They were not vaccinating children that were already infected. And then third, it was an oral vaccine. So they gave this vaccine to half the group, and then the placebo was over 2,000 children that did not receive this vaccine. And they followed these children for 3 years. And they found that in the children that received the placebo, they had a much higher rate of acquiring new H. pylori infections compared to children that had received the vaccine. One opportunity from this study, I think in future studies, is that it seemed like that the efficacy did seem to wane. Whether or not you could administer a booster vaccine at these later time points to improve efficacy later on is still in question. And also, the vaccine, even though it was oral, the children had to take large amounts, 80 mils, for 3 doses. So optimization of that, I think, also would be important in the future. So I'll go ahead and conclude. Gastric cancer is a complex human disease like many others. It's multifactorial, and its pathogenesis really combines the effects of microbial, host, and environmental factors. And I think right now, test and treat strategies for the indiscriminate elimination of H. pylori are not supported by current data unless there's a defined risk factor. So again, thank you so much for the very kind invitation. It's incredible to be at such a wonderful medical university and to see good friends from the past, and look forward to meeting new colleagues as I go forward. I'll be glad to take any questions, and I forgot to say I have no disclosures. So thank you so much.